So we're very fortunate to have Martin Kropff here to give us a presentation with his deep expertise. I remember almost as a PhD student meeting Martin for the first time, and he actually said, well, we've been thinking about G by E by M for a long time. So I'm sure he'll have many excellent comments from his own research activities, but also from his deep involvement in the CG centers and particularly CIMMYT. So looking forward to hearing what you have to say, Martin. I'll hand it over to you.
Thanks, Mark. Wonderful to be here and wonderful to be invited. GBEBM has been something in my whole career that played a role, although I was involved in management as well, but managing science means that you're still involved in the science yourself. Just wondering, how many people here are working in the CGIAR? Limited. There is a great opportunity. I will come back to that later on, because in the CGIAR, I'm retired from the CG officially, but still Emeritus DG of CIMMYT, and we really in the CGIAR need that international collaboration more than ever. And the challenges, Richard showed it and Fred showed it, the moment right now is really ready. There are data, more data than we can handle. There are novel tools like AI. Crop models have not been so much developed. Some great initiatives like with CNU, bringing the crop component in there. But one of my conclusions is also that we need more physiology, understanding. Fred also indicated it very well. You can statistically say, G by E, this is how it's working, but we need to know why to get beyond where we are. So I worked in, you see me here, this is the laser. As a young scientist here, many people from those days don't recognize me anymore, but because I lost my mustache, right? And but here we worked with a great team globally in Asia, with 14 teams in 9 Asian countries on rice modeling. G by E at CIMMYT. This is CIMMYT 50 years old. Great time. A lot of people working on G by E by M, and now I'm involved in GxExM and in the Jan Ingenhouse Institute, working on photosynthesis. But and of course, all my career I've been also linked to Wageningen. Now, the key of the whole thing, basically, in the CG and for all of us is there are many things on the television today that are not related to this, but this is the long-term biggest challenge ever. To contribute to agri-food systems transformation, they have to be transformed for affordable, sufficient, and healthy diets produced within planetary boundaries. It's one sentence that I came up with in CIMMYT. It covers everything almost, and it's enormous. Because basically, and I don't include those slides, but we all know the problem, we get 2 billion more people, where? South Asia and in Africa. We have 800 million people hungry every day, where? South Asia and Africa. Climate change, where is the yield going down and where is the yield going up, for example, for the big crops like wheat and maize? In Africa, South Asia, yields are going down. We've seen it with the models. And in Europe, they go up a little bit. So that's, I call it a perfect storm. So we need all the efforts, not only of the national systems in Africa and South Asia, not together with the CGIAR and FAO and so on, but also with international universities and experts like you all are. So genetics management and governance, that's what is necessary. CIMMYT, we are in all the exciting but also difficult countries where we are working, where we have teams, and we work with more than 300 partners, about 1,500 staff, and in the whole CGIAR it's about 10,000 staff. By far not enough for the challenge that we have. So basically the impact, and that's important to know in the CG, because I'm always interested in what is going to the farmers. What is the benefit of the farmers from the work we do? Science is exciting, but the farmers need that benefit. And the benefit of CIMMYT has been estimated now at $3 to $4 billion extra income annually for smallholder farmers. But we have 500 million of them globally. So we have to keep on going in that sense. But so there's a good start, I would say, but we need all the effort together. And Fred and Richard also indicated that collaboration is necessary. Last week, 2 weeks ago, we had a GxExM conference, 2 days. I had now the time, so I was there for 2 days, and I've seen that everybody is still working and sitting in the sessions on their own interest. And that's what's exciting about this symposium, that hopefully you all are in the statistics session, in the more physiology, and more in the crop modeling sessions. Now, this is basically the challenge in a simple way. These are the world average yields of wheat and maize, 2 of the 3 major crops in the world. So we have seen a major genetic gain. My colleagues call it genetic gain. I always said it's genetic gain and agronomic gain. Let's be honest, right? But this is the average yield going up from 2 tons per hectare to almost 5 tons. That's great, but the challenge is such with the growth of population, it has to grow faster than ever. And basically we have a couple of problems. New diseases coming in all the time in a bigger way. Water, nutrient and energy scarcity is a big issue. It's on television every day, and then climate change is hitting it as well. So through breeding and agronomy together, we have to lift it up. Is it possible? It is possible. Is it simple? No, it's not simple. But how can we then link all the different types of work that all you guys are doing? I will give you a few examples. So this is a slide I used on a modeling conference, because many people are running models, crop models, but how many people know what's in the model and can add new physiological functions in the model? Who is here working with crop models? Quite a few. Who is also changing the model? Of course, Mark. Quite a few. And we need more and more. Please, also when you use crop models, make sure you know what's in there, because then the number of applications go up. You can analyze your data. So you can also not just statistically see what's going on, but why and what is the basis of the G/E/M. You can generate new hypotheses, study emerging behavior that we don't understand, but bringing into a crop model, you can see it. Of course, with breeding and value change, you can work with them, and we have a lot of applications, and that's where it's all being used. And that's also dangerous sometimes, because the models have not been, there's not a real system. So it has its limitations, so you know what you're doing. Yield gap analyses, climate change, regional scenarios, etc., and assisting breeding in G/E/M. I haven't seen in the CGIAR in the last 10 years, not enough of this. The breeders are always, not always, the breeders are very often having their own thing, the physiologists are having their own programs, and then the modelers have their own thing as well. So we need really to bring that together. So this is, for example, a great modeling study that Kai Sonde did at CIMMYT, basically showing with the crop models, and I think he used the DSET models, but also maybe APSIM, the impact of climate change on maize production in Africa. G/E/M. Now, the component of the environment can be done very well with the model, especially when you talk about production, potential production. But here you see the model already. The yellow area here, that's where maize has no future. So we know already that in many areas with the current model scenarios of the climate change models, that we have already areas where the maize cannot grow anymore. And that's happening for many more crops. And of course, with better breeding, we can solve some of the problems. And that's why we need more insight. This is a dream that we had quite a long time ago. It's very simple. You have the crop performance with crop models. You have the physiology. And when you can link the physiology with genotypes, then you can do the genotyping, and then you know exactly how the crop performs. This looks simple, but what physiology is in the model and the genetic coefficients we have in the crop models, that's not so much yet. Especially when you start talking to the mechanistic models of the genomics and transcriptomics people, then you realize that it's not that simple. We made this nice scheme. You can make it even more complicated. We work with Yin Xinyu on it. Ian is also here. And the role of XR, AI, that's where GxExM is focusing on, that's going to be a major difference. Although we had great explanations about AI, and I hope that everybody also here has that knowledge. What can you do with AI and what can you not do with AI? So this is, for example, a mechanistic model of Richard Immink, basically on simply the prediction of genomic data to the moment of flowering. That's the most important thing in cereals, for example, in the crop models, the moment of flowering. Now, I saw Fred's presentation, so you guys are used to these type of slides, right? Normally, I never use such type of slides, to be honest. But this is basically, there are 300 genes involved in the moment of flowering. Now we do it in Arabidopsis. I keep on asking, let's look at the crops, because we have so many data on crops. But in Arabidopsis, we have mutants and all kind of options. So we need to understand it. But what they found now is that it's simpler. It's only 11 key regulators that determine flowering. But still then, this is only one component in a crop model. How can we link this information, and this AP1 seems to be the key component determining the moment of flowering, how can we predict flowering based on this information? And how can we link all this with genomic data, with high throughput systems, and we have all that stuff, but that's where the challenge is right now. In the crop models, and that's what I would like to understand, in crop models we just use a temperature sum. When, of course, photo period is not in there, and that works very well. How come that it's so complex? So how can we link that complexity to the simplicity of what happens in the field? These are scientific challenges that we've seen never before. And of course, nice, also some equations, Fred. But this is an old slide from work that I did with Xin in rice. This is how crop models can help breeders. For example, in completely different environments in Asia, basically we predicted with the Oriza model we developed the potential yield with different periods to flowering. So different temperature sums, different coefficients for the moment to flowering. And you see basically that also this is strange, so you need to go back into the model what's going on, but in most cases there is a sort of optimum duration to flowering, according to the model. And then we looked what the breeders are normally selecting without knowing why, with all respect for breeders, but not knowing exactly the details. And you see that the breeders, that's the dotted lines, are very close to what the model came up with. And that's interesting. If you do it the other way around, so then you can help the breeders finding optimal days to flowering, especially in the tropics where you have 3 crops a year. So these are type of applications that we need much, much more. And this is from Richard, he gave me, because I got excited about AI, he said, Martin, be careful. Be careful. So basically, is it a solution and for what? And basically, he said, make sure that you don't make it bigger and bigger and bigger and collect data that you don't know what the data are about. So make sure that you have well-designed experiments, collect the data that you need, and then with AI you can make sense out of it. So be careful here. Then something about the CG, the breeding and genetics, better and resilient varieties for many traits. Breeders with us at CIMMYT basically focus on specific traits. Drought tolerance, increased yield, biofortification, all kind of components, and they're super successful. So the largest public breeding is in maize and in wheat, and comparable of the level of quality to the private sector, according to private sector people, but at least the sharing of seeds globally, public shared seeds, globally across the world, from land races in the gene banks, is basically something like 80%. So that's why you guys have to work with the CG people, because there's many not yet in the CG. This is, for example, at CIMMYT, a picture I made one day when they were sending material all over the world. 500,000 packages of seeds are being sent to all these locations of wheat and maize from CIMMYT. So all these breeders have networks, they work together, they do a lot of things, and they need top people working on G/E/M. There are good people in there, but they need, I think, more input there as well. And they have great international testing sites where they have hotspots, where they test all the varieties, 60,000 starting in Obregon every year. They test them for heat stress, for wheat blast there, for rust, etc., etc. So these people have a network that is really wonderful. And then this is something that many G/E people don't believe, but they have happy gene packages, they call it. So these are gene packages that apparently work in all those environments, because the breeders, they get all the material and they select the same packages, the same sort of lines in those environments. Why? That's what we need to know. More research is necessary. Yeah, and then host plant resistance, I didn't believe it could be, but basically we need breeders to work faster and faster, and that's what they do. These are 3 major pest and diseases that were there in the time that I was at CIMMYT. Wheat blast, Bangladesh in 1 year, 25% of the yield gone. And the wheat looked good, but there was nothing in there. Fall armyworm all over Africa in the last 5-6 years, killing the maize basically. And maize lethal necrosis, 2 viruses coming in Kenya, 25% of the yield in 1 year gone, and farmers couldn't grow maize anymore. The breeders have been, in all the 3 cases, been able to find in 3 to 4 years host plant resistance, which is really what we need. The same with drought tolerance. As a physiologist, I didn't believe it could be. My people in CIMMYT, they found drought-tolerant maize. This is Margaret, a young farmer's daughter, and here you see 1 crop, but basically the whole crop of the whole plant, of the whole plot. So it's really working. Then I asked the breeders, do you know why? Why? What's happening? They didn't know. So this is where we need the physiology working together with the breeders. And this is the most exciting thing. This is completely new from Dr. Prasanna. He's leading the maize team at CIMMYT. And they have good people, but right now they have already 11 years of data. Then you call, Fred talked about more locations. They have 2,000 locations. Farmers locations and station locations all over mainly Eastern Africa and Southern Africa. They measure everything on even gender and all kinds of things. They have very precious data, and they have also target population of environments defined. Now, in all the CG, that's also novel. And so basically, different types of farms, so well-off farms, resource-constrained farms, etc. And here are all the typologies. And so they have all that information together, and then with GxExM studies, there's so much that can be done. They worked on it. They found already genetic gains in the last 11 years of 2.5 to 1%, depending on the types of varieties. So there is a lot of data there. And also Prasanna told me, I had a call with him last week, that he also would like to work together with people that can add more information to the data. So the CGIAR, I would say, a dream collaboration for modelers, statistical, but also physiologists, and especially also crop modelers. 10,000 people studying crops worldwide, not in 1 location, but in many locations, for wheat and maize and rice as well. AI, new modeling approaches, great opportunities. Modeling expertise is scattered. So I would say we have a situation of data in search of modelers. We had a long period of time that we didn't train people. We didn't have a lot of students in Wageningen. So it's really necessary that more people are able to understand and using those models. Now, the data sets are there, I just showed it, but also understanding the model principles. When you use a crop model, know what's in that model. And then what Ravi Singh, he's now also retired, but the breeders in wheat in CIMMYT told me also, can the modelers identify parents for us breeders to target G2E? Simple question. I asked it at a modelers conference 5 years ago. I didn't get 1 response. Very disappointing. So if people are interested, the breeders are ready because they get all that information from genomics and modelers. They would like to see what kind of crosses should I make, because they have 135,000 accessions of wheat in the gene bank and 34,000 accessions of maize. Which one to choose? What to do? That's where they need your input. And then, of course, we have the crop XR. I'm working on potato and all kinds of crops in the Netherlands, vegetables. We are also bringing that together. And last week I saw that, especially many PhD students, that they sit in each other's sessions. And that's where I think the novel innovations will come from. And then, of course, Jan Ingenhaus will also be presented by Tom, I think, later on this week, where we really have a new institute in Wageningen, funded by 2 private funders, 50 million, to see, can we they said double photosynthesis, but can we enhance photosynthesis? So far, nothing happened. So this is also a challenge, and you will hear more about it today. So to conclude, basically, statistical approaches, analytical models, and new links, let's bring them together. Ecophysiological process-based modeling, improve the genotype components, but Xin-Yu knows more about it. AI for linking genetic physiological traits to crop modeling. GxExM and Jan Ingenhaus are here, willing to work with you. A lot of opportunities, and also the new phenotyping tools. I've seen them now in NPAC here, but also in Utrecht last week. Great opportunities, but make sure that you do the things there that are necessary. And then team genetics breeders, physiologists, and modelers work together. Thank you. Thanks.
Thank you for the inspiring sort of big picture overview and laying out some of the challenges and the motivations for bringing this sort of experiment on a very diverse community of practice together to tackle some of these issues. Any questions that anybody would like to ask Martin, given you've got this great opportunity?
It's a silent group, huh? Hi. I've seen great examples. I think Matthew is not here.
Can you repeat the question?
Huh?
Repeat the question.
Our breeders and physiologists working together, do you have examples of it, right? And Matthew will speak, I think, Matthew Reynolds. He's not here?
End of the day, he will talk online.
Okay, he will talk online. He is one of those physiologists working with breeding lines, and in the end also working together with the breeders. But breeders very often also have their own program. So to make a program really in an integrated way is not that simple. But for example, one great thing that we found, because they had at a given stage all the new lines of wheat, 1 ton per hectare more in Obregon. And I said, why? And they couldn't tell me. And so Matthew and his team, they went, they went to search also physiological traits, and they found very exciting that it was a type of zinc limitation. So basically more grains, more kernels, and you can fill longer in the wheat. So that's also where physiologists and breeders start working together. But it's, to be honest, what I've seen with all breeders and physiologists and so on, they live in their own world. Many scientists are disciplinary. And so that's a big challenge, and AI and all a symposium like this can bring those teams together. But there is a wish, but then the breeders say, but I would like to have an example that they can give me, how should I select the parents for my crossings? So that's also, we need to show as modelers something to the breeders that we can help them.
So Carlos Messina will be joining us over the next 2 days. He would be a really interesting person for you to talk to. So he's a physiologist that I know has driven maize production in the commercial world onto more than 40 million hectares of maize worldwide through the commercial world in North America, South America, and Europe. So he's a physiologist that actually has driven that impact. So you should enjoy having a good discussion with him. He'll be here. So be a good opportunity for you to have that discussion there. Any other questions? One last question, yep.
All right, so Martin, I was thinking of sort of a comment actually from Dijkstra. I was thinking of your happy gene package, the slide that you showed, and then I was eyeballing also that I think most of the environments that you, or the countries you showed, are countries which do not necessarily have access to high technology and probably harsh environment. And it is already known that, it is known that when you raise the mean in a certain trait, you also decrease the environmental sensitivity. And then those environments probably, they sort of selected gene packages which carry genes for environmental sensitivity. So I was wondering, could it explain a little bit that why those happy genes are working in those environments?
Yeah, maybe in the future we can have a microphone for sharing in the room. Oh, we have one to throw around. But basically, it's a happy gene package, what's in there, and how come? And the environments, I've showed the environments, so basically, these are harsh environments, right? Including Australia. Is that maybe the reason? Wheat in India is irrigated very often as well. So I wouldn't say it's all harsh environments. These are good environments as well. But I asked that question because I'm also trained as a physiologist, and as a crop modeler, you have to understand the physiology. So, but I didn't see the answers, and that's I think where breeders can really be helped to be even more successful. They have been super successful, of course. Big applause for them. But basically, they can be more successful if we start understanding that. But for example, with the wheat in Obregon with 1 ton per hectare, all the variety, 1 ton per hectare, more, basically just because of a bigger sink. And I think sink size, I worked on source-sink relations in rice, but this is very complicated and not many people have worked on it. But also, I think the stay green that you just referred to in the maize is also one related to the sink. Anyway, I'm here at least today for quite some hours, and if you're interested or so, because I think this is a great opportunity. I see so many young people, which is also wonderful.
Yeah.
Thank you.
Thank you very much, Martin. Okay, I will move on to our next speaker.